Chapter 8, the nervous system. Most of us can probably remember being told when we were children not to touch the stove or some other source of potential harm. Because children are curious, such warnings often go unheeded. The result, touching a hot stove brings about an immediate response of pulling away and vivid memory of painful fingers. The simple and familiar experience illustrates the functions of the nervous system. 1. To detect changes and feel sensation. 2. To initiate appropriate responses to changes. 3. To organize information for immediate use and store it for future use. The nervous system is one of the regulating systems, in parentheses. The endocrine system is the other and is discussed in chapter 10, close parentheses. The electrochemical impulses of the nervous system make it possible to obtain information about the external or internal environment and do whatever is necessary to maintain homeostasis. Some of this activity is conscious, but much of it happens without our awareness. Nervous system divisions. The nervous system has two divisions. The central nervous system, CNS, consists of the brain and spinal cord. The peripheral nervous system, PNS, consists of cranial nerves and spinal nerves. The PNS includes nerves to and from skin and skeletal muscle. It also includes the autonomic nervous system, ANS, with nerves to visceral effectors, and the enteric nervous system, which is located on the wall of the alimentary tube. The peripheral nervous system relays information to and from the central nervous system, and the brain is the center of activity that integrates this information, initiates responses, and makes us the individuals we are. Nerve tissue. Nerve tissue was briefly described in chapter 4. So we will begin by reviewing what we already know and then add to it. Nerve cells are called neurons or nerve fibers. Whatever their specific functions, all neurons have the same physical parts. The cell body contains the nucleus and is essential for the continued life of the neuron. As you will see, neuron cell bodies are found in central nervous system or close to it in the trunk of the body. In these locations, cell bodies are protected by bone. There are no cell bodies in the arms and legs, which are much more subject to injury. Dendrites are processes, in parentheses, extensions that transmit electrical impulses toward the cell body. The one axon of a neuron transmits impulses away from the cell body, perhaps to another neuron or to an effector cell of muscle or glandular tissue. It is the cell membrane of the dendrite, cell body, and axon that carries the electrical nerve impulse. In the peripheral nervous system, axon and dendrites are wrapped in specialized cells called Schwann cells. During embryonic development, Schwann cells grow to surround the neuron processes, enclosing them in several layers of Schwann cell membrane. These layers are the myelin sheath. Myelin is a phospholipid that electrically insulates neurons from one another. Without the myelin sheath, neurons would short circuit, just as electrical wires would if they were not insulated. The spaces between adjacent Schwann cells or segments of myelin sheath are also called nodes of Ranvier, in parentheses, neurofibril nodes, close parentheses. These nodes are the part of the neuron cell membrane that depolarize when an electrical impulse is transmitted, and their presence increases the speed of transmission. The nuclei and cytoplasm of Schwann cells are wrapped around the outside of the myelin sheath and are called neurolema, which becomes very important if nerves are damaged. If a peripheral nerve is severed and reattached precisely by microsurgery, the axons and dendrites may regenerate through the tunnels formed by the neurolemas. The Schwann cells are also believed to produce a chemical growth factor that stimulates regeneration. Although this regeneration may take months, the nerves may eventually reestablish their proper connections, and the person may regain some sensation and movement in the once severed limb. In the central nervous system, the myelin sheaths are formed by oligodendrocytes, one of the neuroglia, in parentheses, glia cells, close parentheses. The specialized cells found only in the brain and spinal cord. Because no Schwann cells are present, however, there is no neurolema, and regeneration of neurons does not occur. This is why severing of the spinal cord, for example, results in permanent loss of function. Microglia are another kind of neuroglia. These cells are phagocytes and are constantly moving. They clean up cellular debris and damaged cells and phagocytize pathogens. Yet another type of glia cell is a astrocyte, literally star cell. In the embryo, these cells provide a framework of the migrating neurons that will form the brain. Thereafter, the extension of the astrocytes are wrapped around neurons and around brain capillaries. And these cells are not passive supports. Astrocytes help regulate localized blood flow within the brain. They are believed to detect the release of neurotransmitters by neurons and to increase blood flow when their neurons become more active. Astrocytes also contribute to the blood-brain barrier, which prevents potentially harmful waste products in the blood from diffusing out onto the brain tissue. These waste products are normal in the blood and tissue fluid, but brain tissue is much more sensitive to even low levels of them than are other tissues such as muscle tissue or connective tissue. The capillaries of the brain also contribute to this barrier because their cells are tightly packed edge to edge, making brain capillaries less permeable than other capillaries. Some harmful chemicals such as alcohol and nicotine do cross the blood-brain barrier, and the rabies virus is a pathogen that can cross it. A disadvantage of the blood-brain barrier is that some useful medications cannot cross it, and the antibodies produced by the lymphocytes cross only with difficulty. This becomes an important consideration when treating brain infections or other diseases or disorders. Takeaways. 1. In which divisions of the nervous system, divisions of the nervous system are the cell bodies of neurons found? Why there? 2. Which of the neuroglia are phagocytic? What do they eat? Which of the neuroglia contribute to the blood-brain barrier? Answer. 1. Cell bodies of neurons are found in the CNS or in the PNS within the skull and trunk of the body, where they are protected by bone. 2. Microglia phagocytize cell debris and pathogens. The astrocytes contribute to the blood-brain barrier. The nerve impulse. The events of an electrical nerve impulse are the same as those of the electrical impulse generated in muscle fibers, which is discussed in Chapter 7. Stated simply, a neuron not carrying an impulse is in the state of polarization, with Na plus ions more abundant outside the cell and K plus ions and negative ions more abundant inside the cell. The neuron has a positive charge on the outside of the cell membrane and a relative negative charge inside. A stimulus, such as a neurotransmitter, makes the membrane very permeable to Na plus ions, which rush into the cell. This brings about depolarization, a reversal of charges on the membrane. The outside now has a negative charge and the inside has a positive charge. As soon as depolarization takes place, the neuron membrane becomes very permeable to K plus ions, which rush out of the cell. This restores the positive charge outside, and the negative charge inside is called a repolarization. The term action potential refers to depolarization followed by repolarization. Then the sodium and potassium pumps return, Na plus ions outside and K plus ions inside, and the neurons is ready to respond to another stimulus and transmit another impulse. An action potential in response to a stimulus takes place very rapidly and is measured in milliseconds. An individual neuron is capable of transmitting hundreds of action potentials, in parentheses, impulses, close parentheses, each second. A summary of the events of nerve impulse transmission is given in Table 8.2. Transmission of electrical impulses is very rapid, especially if the neurons are myelinated. The presence of an insulating myelin sheath increases the velocity of impulses by making the neurons shorter. Since only the nodes of Ranvier depolarize, look back at the motor neuron in Figure 8.1 and mentally add up the yellow spaces between the green Schwann cells. In total, they are very short, and this type of transmission is called saltatory conduction. Many of our neurons are capable of transmitting impulses at a speed of many meters per second. Imagine a person 6 feet, in parentheses, about 2 meters, close parentheses, tall, and stubs a toe. Sensory impulses travel from toe to the brain in less than a second, crossing a few synapses along the way, and although diffusion across synapses is slow, the synapses are so small that this does not significantly affect the velocity of impulses in a living person. Close parentheses. Electrical impulses enable the nervous system to communicate very rapidly with all parts of the body, making it such an important regulatory system. You may have read or heard that nerve impulses can be called messages, as in sensory neurons send messages to the brain, or the brain sends messages to muscles. To understand how the nervous system works, however, this may not be the best analogy. A message says something. Emails, letters, or phone calls say something specific and rarely the same. Nerve impulses, however, are all exactly the same electrical action potentials. The effect of a nerve impulse is not a quality of the impulse itself, but rather of its destination. For example, when a person takes a step, nerve impulses from the left foot arrive in the cutaneous area for the left foot in the right parietal lobe of the cerebrum, and the person feels the left foot hit the floor. If those impulses could be routed to the area for the right foot in the left parietal lobe, in parentheses, this type of experiment has been done with rats, close parentheses, we would feel as if the right foot were hitting the floor instead the left. Destination is all that matters. Recipients is all that matters. Just as the electricity, all the same, carried by the wires of a house can enable different appliances to fulfill their purposes, so too can nerve impulses enable bodily functions. Impulses arrive at sweat glands. Impulses arrive at sweat glands, bring about secretion of sweat, because that is what these glandular cells do. Impulses arriving in the visual area of the occipital lobe bring about a sensation of seeing something, because that is what these neurons do. Impulses arriving at a skeletal muscle cells bring about contraction, because that is what muscles cells do. Therefore, a nerve impulse is not in itself a message. If anything, a nerve impulse may be considered more like a switch or simple signal, in that it activates or inhibits the cell, tissue, or organ of its destination. In keeping with this idea, the brain may be likened to the conductor of an orchestra. And let us say the musicians are the skeletal muscles, in parentheses, of the legs, trunk, and arms, close parentheses, needed to climb a mountain. Each muscle is structured so as to do a particular job. As each musician is trained on a specific instrument, the conductor does not play the basson or trumpet or timpani or cello, but will cue the musicians to play their parts. These cues will also signal the proper time for each and in the proper sequence. For musical activity, this would be coordination. The cues by the conductor, the signals, are very much the same. The signals are not the effect. The recipients of the signals create the effect. On their way to their recipient or effector, cells, electrical nerve impulses. On the way to their recipient cells, electrical nerve impulses cross one or more synapses. You may recall from chapter 4 that at the synapse, nerve impulse transmissions change from electrical to chemical and depends on the release of neurotransmitters. Synapses. Neurons that transmit impulses to other neurons or to the effector cells do not actually touch one another. The small gap or space between the axon of one neuron and the dendrites or cell body of the next neuron is called the synapse. Within the synaptic knob, in parentheses, terminal end, close parentheses, of the presynaptic axon is a chemical neurotransmitter that is released into the synapse by the arrival of an electrical nerve impulse. The neurotransmitter diffuses across the synapse, combines with specific receptor sites on the cell membrane of the postsynaptic neuron, and there generates an electrical impulse that is, in turn, carried by this neuron's axon to the next synapse, and so forth. A chemical inactivator at the cell body or dendrite of the postsynaptic neuron quickly inactivates the neurotransmitter. This prevents unwanted continuous impulses unless a new impulse from the first neuron releases more neurotransmitter. Many synapses are termed excitatory because the neurotransmitter causes the postsynaptic neuron to depolarize, in parentheses, become more negative outside as Na plus ions enter the cell, close parentheses, and transmit an electrical impulse to another neuron, muscle cell, or gland. Some synapses, however, are inhibitory, meaning that the neurotransmitter causes the postsynaptic neuron to hyperpolarize. Hyperpolarization means that the neuron membrane becomes even more positive outside as K plus ions leave the cell or Cl ions enter the cell, and therefore the neuron does not transmit an electrical impulse. Such inhibitory synapses are important, for example, for slowing the heart rate and for balancing excitatory impulse transmitted to skeletal muscles. With respect to the skeletal muscles, this inhibition prevents excessive contraction and is necessary for coordination. One important consequence of the presence of synapses is that they ensure one-way transmission of impulses in a living person. A nerve impulse cannot go backward across a synapse because no transmitter is released by the dendrites or cell body. Neurotransmitters can be released only by a neuron's axon, which does not have receptor sites for it, as does the postsynaptic membrane. Keep this in mind when we discuss the types of neurons later in this chapter. An example of a neurotransmitter is acetylcholine, which is found at neuromuscular junctions. In the CNS and in much of the peripheral nervous system, acetylcholine usually makes a postsynaptic membrane more permeable to Na plus ions, which brings about depolarization of the postsynaptic neuron. Cholinesterase is the inactivator of acetylcholine. There are many other neurotransmitters, especially in the central nervous system. These include dopamine, GABA, norepinephrine, glutamate, and serotonin. Each of these neurotransmitters has its own chemical inactivator. Some neurotransmitters are reabsorbed into the neurons that secreted them. This process is called reuptake and also terminates the effect of the transmitter. The complexity and variety of synapses make them frequent targets of medications. For example, drugs that alter mood behavior often act on specific neurotransmitter in the brain, and antihypertensive drugs often synapse transmission at the smooth muscle of blood vessel. Takeaways 2. 1. How is depolarization the same for a neuron cell membrane and a sarcolemma? Compare in terms of electrical charges and movement of ions. Answer. The reversal of charges is the same for both during depolarization from positive to negative outside the membrane and from negative to positive inside the membrane. The movement of ions is also the same. Na plus ions enter the cell. Takeaways 2. How is synaptic transmission the same between neurons at the neuromuscular junction? Answer. Synaptic transmission is the same in that a neurotransmitter is released, crosses the synapse, and generates an impulse in the receiving cell. Types of neurons. Neurons may be classified into 3 groups: sensory neurons, motor neurons, and interneurons. Sensory neurons, or afferent neurons, carry impulses from receptors to the central nervous system. Receptors are structures that detect external or internal changes and generate impulses that travel along afferent neurons to the CNS. The central nervous system interprets these impulses as a sensation. Sensory neurons from receptors in skin, skeletal muscle, and joints called somatic. Those from receptors in internal organs are called visceral sensory neurons. Motor neurons, in parentheses, or efferent neurons, close parentheses, carry impulses from the central nervous system to effectors. The 2 general types of effectors are muscles and glands. In response to impulses, muscles contract or relax and glands secrete or do not. Motor neurons linked to skeletal muscles are called somatic. Those to smooth muscles, cardiac muscle, and glands are called visceral. Sensory and motor neurons make up the peripheral nervous system. Visceral motor neurons form the autonomic nervous system, a specialized subdivision of the PNS that will be discussed later in this chapter. Interneurons are found entirely within the central nervous system. They are arranged so as to carry only sensory or motor impulses, or to integrate these functions. Some interneurons in the brain are concerned with thinking, learning, and memory. A neuron carries impulses in only one direction. This is the result of the neuron structure and location, as well as its physical arrangement with other neurons and the resulting pattern of synapses. The functioning nervous system, therefore, is an enormous network of one-way streets, and there is no danger of impulses running into and canceling one another out. Nervous and nerve tracts. A nerve is a group of axons and or dendrites of many neurons with blood vessels and connective tissue. Sensory nerves are made only of sensory neurons. The optic nerves for vision and olfactory nerves for smell are examples of nerves with a purely sensory function. Motor nerves are made only of motor neurons. Autonomic nerves are motor nerves. A mixed nerve contains both sensory and motor neurons. Most of our peripheral nerves, such as the sciatic nerves in the legs, are mixed nerves. The term nerve tract refers to a group of neurons within the central nervous system. All the neurons in a nerve tract are concerned with either sensory or motor activity. These tracts are often referred to as white matter because the myelin sheaths of the neurons give them a white color. This term is used for parts of both the brain and the spinal cord. Takeaways 3. 1. What are the components of a mixed nerve with respect to the CNS? What does each component do? Answer. A mixed nerve consists of both sensory and motor neurons. Sensory neurons transmit impulses to the CNS from receptors. Motor neurons transmit impulses from the CNS to effectors. 2. What are nerve tracts? Where are nerve tracts found? They may be referred to as fill in the blank. Answer. Nerve tracts are found in the CNS and are called the white matter of the brain and spinal cord. The spinal cord transmits impulses to and from the brain and is the integrating center for the spinal cord reflexes. Although this statement of functions is very brief and sounds very simple, the spinal cord is of great importance to the nervous system and to the body as a whole. Enclosed within the vertebral canals and the meninges, the spinal cord is well protected from mechanical injury. In length, the spinal cord extends from the foramen magnum of the occipital bone to the disc between the first and second lumbar vertebrae. A cross-section of the spinal cord is shown in Figure 8.3. Refer to it as you read the following. The internal gray matter is said to be shaped like the letter H or a butterfly. The wings of the butterfly are called horns. Dorsal, in parentheses, or posterior, close parentheses. Lateral and ventral, or anterior. The gray matter consists of cell bodies of motor neurons and interneuron. It is made of myelinated axons and dendrites of interneurons. These nerve fibers are grouped into nerve tracts on the basis of their functions. Ascending tracts such as the dorsal columns and spinothalamic tracts, close parentheses, carry sensory impulses to the brain. Descending tracts, in parentheses, such as the corticospinal and rubrospinal tracts, close parentheses, carry motor impulses away from the brain. Lastly, find the central canal. This contains cerebrospinal fluid and is continuous with cavities in the brain called ventricles. There are 31 pairs of spinal nerves. Those that emerge from the spinal cord. The nerves are named according to their respective vertebrae. 8 cervical pairs, 12 thoracic pairs, 5 lumbar pairs, 5 sacral pairs, and 1 very small coccygeal pair. These are shown in Figure 8.4. Notice that each nerve is designated by a letter and a number. The 8th cervical nerve is C8. The first thoracic nerve is T1, and so on. In general, the cervical nerves supply the back of the head, neck, shoulders, arms, and diaphragm. The first thoracic nerve also contributes to nerves in the arm. The remaining thoracic nerves supply the trunk of the body. The lumbar and sacral nerves supply the hips, pelvic cavity, and legs. Notice that the lumbar and sacral nerves hang below the end of the spinal cord, in parentheses, in order to reach their proper openings to exit from the vertebral canal, close parentheses. This is called the quota equina, literally the horse's tail. Some of the important peripheral nerves and their destinations are listed in Table 8.3. Each spinal nerve has 2 roots, which are neurons entering or leaving the spinal cord. The dorsal root is made of sensory neurons that carry impulses into the spinal cord. The dorsal root ganglion is an enlarged part of the dorsal root that contains the cell bodies of sensory neurons. The term ganglion means a group of cell bodies outside the CNS. These cell bodies are within the vertebral canal and are thereby protected from injury. The ventral root is the motor root. It is made of the axons of motor neurons carrying impulses from the spinal cord to muscle glands. The cell bodies of these motor neurons, as mentioned previously, are in the gray matter of the spinal cord. Somatic motor cell bodies are in the ventral horns, and the autonomic cell bodies are in the lateral horns. When the two nerve roots merge just before entering the backbone, the spinal nerve thus formed is a mixed nerve. Spinal cord reflexes. When you hear the term reflex, you may think of an action that just happened, and in part this is so. A reflex is an involuntary response to a stimulus, that is, an automatic action stimulated by a specific change of some kind. Spinal cord reflexes are those that do not depend directly on the brain, although the brain may inhibit or enhance them. We do not have to think about these reflexes, which is very important, as you will see. Reflex arc. A reflex arc is the pathway that nerve impulses travel when reflex is elicited, and there are 5 essential parts. 1. Receptors. Detect a change, stimulus, and generate impulses. 2. Sensory neurons. Transmit impulses from receptors to the CNS. 3. Central nervous system. Contains one or more synapses. In parentheses, interneurons may be part of the pathway. Close parentheses. 4. Motor neurons transmit impulses from the CNS to the effector. 5. Effector performs its characteristic action. Let us now look at the reflex arc of a specific reflex, the patellar or knee-jerk reflex, with which you are probably familiar. In this reflex, a tap on the patellar tendon just below the kneecap causes extension of the lower leg. This is a stretch reflex, which means that a muscle that is stretched will automatically contract. Refer now to figure 8.5 as you read the following. In the quadriceps femoris muscle 1, stretch receptors that detect the stretching produced by striking the patellar tendon. These receptors generate impulses that are carried along. 2. Sensory neurons in the femoral nerve. 2. 3. The spinal cord. In the spinal cord, the sensory neurons synapse with 4 motor neurons. This is a 2-neuron reflex. The motor neurons in the femoral nerve carry impulses back to 5, the quadriceps femoris, the effector, which contracts and extends the lower leg. The patellar reflex is one of many used clinically to determine whether the nervous system is functioning properly. If the patellar reflex were absent in a patient, the problem could be in the thigh muscle, the femoral nerve, or the spinal cord. Further testing would be needed to determine the precise break in the reflex arc. If the reflex is normal, however, that means that all parts of the reflex arc are intact. So the testing of reflexes may be a first step in the clinical assessment of neurologic damage. You may be wondering why we have such reflexes. These stretch reflexes, what is their importance in our everyday lives? Do they occur? Most of the time we are not aware of them, but if you have ever seen someone fall asleep while sitting, you may have seen 1. The person's head drops and then suddenly snaps up. The posterior neck muscles were stretched by the droop and then contracted, abruptly pulling the head upright. Other stretch reflexes take place less dramatically. Imagine a person standing upright. Is the body perfectly still? No, it isn't, because gravity exerts a downward pull. However, if the body tilts to the left, the right side of the leg and trunk are stretched, and these stretched muscles automatically contract and pull the body upright again. This is the purpose of stretch reflexes. They help keep us upright without our having to think about doing so. If the brain had to make a decision every time we swayed a bit, all our concentration would be needed just to remain standing. Since these are spinal cord reflexes, the brain is not directly involved. The brain may become aware that a reflex has taken place, but that involves another set of neurons carrying impulses to the brain. Flexor reflex. Flexor reflexes or withdrawal reflexes are another type of spinal cord reflex. The stimulus is something painful and potentially harmful, and the response is to pull away from it. If you inadvertently touch a hot stove, you automatically pull your hand away. Flexor reflexes are 3-neuron reflexes because sensory neurons synapse with interneurons in the spinal cord, which in turn synapse with motor neurons. Again, however, the brain does not have to make a decision to protect the body. The flexor reflex does that automatically. The brain may know that the reflex has taken place and may even learn from the experience, but that requires different neurons, not the reflex arc. Takeaways 4. 1. Where are motor neuron cell bodies located? More specifically, where are somatic motor neuron cell bodies located? Sensory neuron cell bodies, axon of interneurons. Sure. Motor neuron cell bodies are located in the gray matter of the spinal cord. Somatic motor neuron cell bodies are in the ventral anterior horns of the gray matter. Sensory neuron cell bodies are located in the dorsal root ganglia just outside the spinal cord. The axons of interneurons are located in the white matter, ascending and descending nerve tracts of the spinal cord. The axons of interneurons are located in the white matter, in parentheses, ascending and descending nerve tracts, close parentheses, of the spinal cord. 2. With respect to a reflex arc, put the following in proper sequence. A. Spinal cord. B. Effector. C. Receptor. D. Sensory neuron. E. Motor neuron. Answer. C. D. A. E. B. 3. With respect to the spinal cord, what do C8 and T2 stand for? Answer. C8 designates the 8th pair of cervical spinal nerves. T2 designates the 2nd pair of thoracic spinal nerves. The brain. The brain is made of approximately 86 billion neurons. This most recent estimate is a bit lower than the previous estimate of 100 billion, and about the same number of glial cells, and contains trillions of synapses, all of which function as an integrated whole. The major parts of the brain are the medulla, pons, and the midbrain, collectively called the brainstem, the cerebellum, the hypothalamus, the thalamus, and the cerebrum. These parts are shown in figure A6. We will discuss each part separately, but keep in mind that they are all interconnected by way of those trillions of synapses and work together. Ventricles. The ventricles are 4 cavities within the brain, 2 lateral ventricles, the 3rd ventricle, and the 4th ventricle. The ventricles are lined with ciliated ependymal cells and filled with cerebrospinal fluid, CSF. Each ventricle contains a capillary network called a porod plexus, which forms the cerebrospinal fluid from blood plasma. Cerebrospinal fluid is tissue fluid of the central nervous system. Its circulation and functions will be discussed in the section on meninges. Medulla. The medulla extends from the spinal cord to the pons and is anterior to the cerebrum. Its functions are those we think of as vital, in parentheses, as in vital signs, close parentheses. The medulla contains cardiac centers that regulate heart rate, vasomotor centers that regulate the diameter of blood vessels and thereby blood pressure, and respiratory centers that regulate breathing. You can see why a crushing injury to the occipital bone may be rapidly fatal. We cannot survive without the medulla. Also in the medulla are reflex centers for coughing, sneezing, swallowing, and vomiting. The pons. The pons bulges anteriorly from the upper part of the medulla. Within the pons are two respiratory centers that work with those in the medulla to produce a normal breathing rhythm. The many other neurons in the pons connect the medulla with other parts of the brain. Midbrain. The midbrain extends from the pons to the hypothalamus and encloses the cerebral aqueduct, a tunnel that connects the third and fourth ventricles. Several different kinds of reflexes are integrated in the midbrain, including visual and auditory reflexes. If you see a wasp flying toward you, you automatically duck or twist away. This is a visual reflex. Keeping an eye on that wasp, actually both eyes, until it flies away is called tracking. The coordinated eye movement that make this possible are also a reflex. Other examples are following the flight of a home run ball or a long touchdown pass. The ability to read depends in part on the coordinated movement of the eyeballs from word to word. This is scanning and is an other reflex mediated by the midbrain. Turning your head, ear to a sound is an example of an auditory reflex, as is the startle. A reflex, a jump or cringe upon hearing a sudden loud sound such as thunder. The midbrain is also concerned with what are called righting reflexes, those that keep the head upright and maintain balance or equilibrium. When a cat falls from a tree and lands on its feet, if it is not up too high, its midbrain helped get its head upright, and other parts of the brain then swing the trunk and legs into place for a landing. Human midbrains are not nearly that efficient. Cerebellum is separated from the medulla and pons by the fourth ventricle and is inferior to the occipital lobes of the cerebrum. As you already know, many of the functions of the cerebellum are concerned with movement. These include coordination, regulation of muscle tone, the appropriate trajectory and endpoint of movements, and the maintenance of posture and equilibrium. Notice that these are all involuntary. That is, the cerebellum functions below the level of conscious thought. This is important to permit the conscious brain to work without being overburdened. If you decide to pick up a pencil, for example, the impulses for arm movement come from the cerebrum. The cerebellum then modifies these impulses so that your arm and finger movements are coordinated and you don't reach past the pencil. The cerebellum seems also to be involved in certain sensory functions. For example, if you close your eyes and someone's placed a tennis ball in one hand and a baseball in the other, could you tell which was which? Certainly, you could by feel of each, the texture and the weight or heft. If you pick up a plastic container of coffee, could you tell that the cup is full, half full, or empty? Again, you certainly could. Do you think about it? No, the cerebellum is in part responsible for disability. Also believed to be a function of the cerebellum is the muscle memory mentioned in the last chapter. A learned physical task from climbing stairs to playing a Beethoven piano sonata can be performed again and again without having to think about each move. Sensation, movement, and memory have been integrated by the cerebellum. To regulate equilibrium, the cerebellum and midbrain uses information about gravity and movement provided by receptors in the inner ears. These receptors are discussed further in chapter 9. Hypothalamus. Located superior to the pituitary gland and inferior to thalamus, the hypothalamus is a small area of the brain with many diverse functions. 1. Production of antidiuretic hormone, ADH, and oxytocin. These hormones are then stored in the posterior pituitary gland. ADH enables the kidneys to reabsorb water back into the blood and thus helps maintain blood volume, relocated to our water volume. Other neurons in the hypothalamus are believed to contribute to the sensation of thirst. Oxytocin causes contractions to the uterus to bring about labor and delivery. 2. Production of releasing hormones, in parentheses, also called releasing factors, close parentheses, that stimulate the secretion of hormones by the anterior pituitary gland. Because these hormones are covered in Chapter 10, a single example will be given here. The hypothalamus produces growth hormone, releasing hormone, GHRH, which stimulates the anterior pituitary gland to secrete growth hormone, GH. Regulation of body temperature by promoting responses such as sweating in warm environment or shivering in a cold environment. Regulation of food intake. The hypothalamus is believed to respond to changes in blood nutrient levels, to chemicals secreted by fat cells, and to hormones secreted by the gastrointestinal tract. For example, during a meal, after a certain duration of digestion, the small intestine produces a hormone that circulates to the hypothalamus and brings about a sensation of satiety or fullness, and we tend to stop eating. Integration of the functioning of the autonomic nervous system, which in turn regulates the activities of organs such as the heart, blood vessels, and intestines. This will be discussed in more detail later in this chapter. 6. Stimulation of visceral responses during emotional situations. When we are angry, heart rate usually increases. Most of us, when embarrassed, will blush, which is vasodilation in the skin of the face. These responses are brought about by the autonomic nervous system. When the hypothalamus perceives a change in emotional state, the neurologic basis of our emotions is not well understood, and the visceral responses to emotions are not something most of us can control. 7. Regulation of body rhythms such as secretions of hormones, sleep cycles, changes in mood or mental alertness. Other parts of the brain contribute, and this is often referred to as our biological clock. The rhythms as circadian rhythms, meaning about a day. If you have ever had to stay awake for 24 hours, you know how disorienting it can be until the hypothalamic biological clock has been reset. Thalamus. The thalamus is superior to the hypothalamus and inferior to the cerebrum. The third ventricle is a narrow cavity that passes through both the thalamus and hypothalamus. Many of these functions of the thalamus are concerned with sensation, sensory impulses to the brain, except those for sense of smell. Follow neuron pathways that first enter the thalamus, which groups the impulses before relaying them to the cerebrum, where sensations are felt. For example, holding a cup of hot coffee generates impulses for heat, touch, and texture, and the shape of the cup, in parentheses, muscle sense. But we do not experience these as separate sensations. The thalamus integrates the impulses from the cutaneous receptors and from the cerebellum. That is, it puts them together in a sort of electrochemical package and directs them to the sensory area in the parietal lobe of the cerebrum, so that the neurons there fill the hole and are able to interpret the sensation quickly. Some sensations, especially unpleasant ones such as pain, are believed to be felt by the thalamus. However, the thalamus cannot localize the sensation. That is, it does not know where the painful sensation is coming from. The sensory areas of the cerebrum are required for localization and precise awareness. The thalamus may also suppress unimportant sensations. If you are reading an enjoyable book, you may not notice someone coming into the room. By temporarily blocking minor sensations, the thalamus permits the cerebrum to concentrate on important tasks. Parts of the thalamus are also involved in alertness and awareness, and others contribute to memory. For these functions, as for others, the thalamus works very closely with the cerebrum. The largest part of the human brain is the cerebrum, which consists of two hemispheres separated by the longitudinal fissure. At the base of this deep groove is the corpus callosum, a band of 200 million neurons that connects the right and left hemispheres. Within each hemisphere is a lateral ventricle. The surface of the cerebrum is gray matter called the cerebral cortex. Gray matter consists of cell bodies of neurons which carry out many functions of the cerebrum. Internal to the gray matter is white matter, made of myelinated axons and dendrites that connect the lobes of the cerebrum to one another and to all other parts of the brain. In the human brain, the cerebral cortex is folded extensively. The folds are called convolutions or gyri, and the grooves between them are fissures or sulci. This folding permits the presence of millions more neurons in the cerebral cortex. The cerebral cortex of an animal such as a dog or cat does not have the extensive folding. This difference enables us to read, speak, and do so many other human things that dogs and cats cannot do. The cerebral cortex is divided into lobes that have the same names as the cranial bones external to them. Therefore, each hemisphere has a frontal lobe, parietal lobe, temporal lobe, and occipital bone. These lobes have been mapped. That is, certain areas are known to be associated with specific functions. We will discuss these functions of the cerebrum according to these mapped areas. Frontal lobes. Within the frontal lobes are the motor areas that generate the impulses for involuntary movement. The largest parts are for movement of the hands and face. Those areas with many muscles capable of very fine or precise movements. It is the large size of the motor area devoted to them that give these muscles their precision. The left motor area controls movement on the right side of the body, and the right motor area controls the left side of the body. This is why a patient who has had a cerebrovascular accident or stroke in the right frontal lobe will have paralysis of muscles on the left side. Anterior to the motor areas are the premotor areas, which are concerned with learned motor skills that require a sequence of movements. Tying shoelaces, for example, seems almost automatic to us. We forget having learned it. It is not a reflex. However, rather the premotor cortex has learned the sequence so well that we are able to repeat it without consciously thinking about it. Parts of the frontal lobe just behind the ears are the prefrontal or orbitofrontal cortex. This area is concerned with things such as keeping emotional responses appropriate to the situation, realizing that there are standards of behaviors and following them, and anticipating and following for the future. An example may be helpful to put all this together. Someone with damage to the prefrontal area might become enraged if a pen ran out of ink during class, might throw the pen at someone, and might not think that a pen will be needed tomorrow and that it's time to go buy one. As you can see, the prefrontal cortex is very important for social behavior. That is the part of the brain that enables us to realize that other people might not always think the way we do. This is the part that thinks, oops, and allows us to apologize for words or actions that perhaps poorly considered. The prefrontal cortex greatly contributes to what makes us human and able to enjoy the humanity of others. Also in the frontal lobe, usually only the left lobe for most right-handed people is Broca's motor speech area, which controls the movement of the mouth involved in speaking. Parietal lobes. The general sensory areas in the parietal lobes receive impulses from receptors in the skin and feel and interpret the cutaneous sensations. The left areas for the right side of the body and vice versa. These areas also receive impulses from stretch receptors and muscles from conscious muscle sense. The largest portions of these areas are for sensation in the hands and face, those parts of the body with the most cutaneous receptors and the most muscle receptors. The taste areas, which overlap the parietal and temporal lobes, receive impulses from taste buds and other receptors on the tongue and elsewhere in the oral cavity. Amen. Temporal lobes. The olfactory areas in the temporal lobes receive impulses from receptors in the nasal cavities for the sense of smell. The olfactory association area learns the meaning of odors, such as the smell of sour milk or fire or brownies baking in the oven, and enables the thinking cerebrum to use that information effectively. The auditory areas, as their name suggests, receive impulses from receptors in the inner ear for hearing. The auditory association area is quite large. Part of it is concerned with the meanings of words we hear, that is, with speech. Other parts are for the interpretation of sounds such as thunder during a storm, an ambulance siren, or a baby crying. Without proper interpretation, we would hear the sound but would not know what it meant or be able to integrate it with other senses and could not respond appropriately. Also in the temporal and parietal lobes in the left hemisphere, for most of us, are other speech areas concerned with the thought that precedes speech. Each of us can probably recall times we have spoken without thinking, but in actuality, that is not possible. The thinking takes place very rapidly and is essential in order to be able to speak. Occipital lobes. Impulses from the retinas of the eyes travel along the optic nerves to the visual areas in the occipital lobes. These areas see. The visual association areas interpret what is seen and enable the thinking cerebrum to use the information. Imagine looking back at a clock. Seeing the clock is far different from being able to interpret it. At one time, we learned to interpret the clock face and hands, and now we do not have to consciously decide what time the clock is showing. We can simply use that information, such as hurrying a bit so as not to be late to class. Other parts of the occipital lobes are concerned with spatial relationships. That is, things such as judging distance and seeing in 3 dimensions, or the ability to read a map and relate it to the physical world. The cerebral cortex has the characteristics of neuroplasticity. To be plastic is to be capable of taking on or being molded into different shapes. For the cortex, it is the ability to adapt to changing needs, to recruit different neurons for certain functions as may occur during childhood or recovery from a stroke. Another example is the visual cortex of a person who is born blind. The neurons in the occipital lobes that would have been used for vision will often be used for another function. Some may become part of the auditory area that is used to localize sound and estimate their distance. Those of us who can see may not rely on hearing for localization. We simply look at where we think the sound came from. A blind person cannot do this and may have an extensive mental catalog of sounds, meanings of sounds, distances of sounds, and so on. Some of these in the part of the cortex that normally is for vision. The younger the person, the more plastic the brain. The brains of children are extraordinarily adaptable. As we get older, plasticity diminishes somewhat, but it is still present. Association areas. As you can see in figure 8.8, Many parts of the cerebral cortex are not concerned with movement or a particular sensation. These may be called association areas and perhaps are what truly make us individuals. It is probably these areas that give each of us personality, a sense of humor, and the ability to reason and use logic. Learning and memory are also functions of these areas. Although much has been learned about the formation of memories, the processes are still incompletely understood and mostly beyond the scope of this book. Briefly, however, we can say that memories of things such as people or books or what you did last summer involve the hippocampus, part of the temporal lobe on the floor of the lateral ventricle. The two hippocampi seem to collect information from many areas of the cerebral cortex. When you meet a friend, for example, the memory emerges as a whole. Here's Fred, not in pieces. People whose hippocampi are damaged cannot form new memories that last more than a few seconds and have difficulty recalling old memories. The right hippocampus is also believed to be involved in spatial cognition. For example, if you are in school and a friend asks you the shortest way to your home, you will probably quickly form a mental map. You can see how much memory that involves, but the hippocampus can take it a step further and make your memories three-dimensional and mentally visible. You can see your way home. That is spatial cognition. It is believed that most, if not all, of what we have experienced or learned is stored somewhere in the brain. That is long-term memory, whether conscious or subconscious, and probably involves the creation of new connections in the brain. Sometimes a trigger may bring back memories from years or even decades ago. A certain scent or a song can act as possible triggers. Then we find ourselves recalling something from the past or wondering where it came from. The loss of personality due to the destruction of brain neurons is perhaps most dramatically seen in Alzheimer's disease. Basal ganglia. The basal ganglia are paired masses of gray matter within the white matter of the cerebral hemispheres. Their functions are certain subconscious aspects of voluntary movement, and they work with the cerebellum. The basal ganglia help regulate muscle tone, and they coordinate accessory movements such as swinging the arms when walking or gesturing while speaking. Gestures and facial expressions can be as eloquent as words and often accompany and complement our conversation. The most common disorder of the basal ganglia is Parkinson's disease. Corpus callosum. As mentioned previously, the corpus callosum is a band of nerve fibers that connects the left and right cerebral hemispheres. This enables each hemisphere to know what the other is doing. The person with musical training has a thicker corpus callosum than someone without. Music apparently makes demands on the cerebrum that non-music study does not, and the cerebrum responds with more neurons and synapses. For all people, even those who are not musicians, the corpus callosum is important because for most of us, the left hemisphere contains speech areas and the right hemisphere does not. The corpus callosum therefore lets the left hemisphere know what the right hemisphere is thinking about, and the right hemisphere know what the left hemisphere is thinking and talking about. A brief example may be helpful. If you put your left hand behind your back and someone places a pencil in your hand, in parentheses, you are not looking at it, and close parentheses, and asks you what it is, would you be able to say? Yes, you would. You would feel the shape and weight of the pencil and find the point and the eraser. The sensory impulses from your left hand are interpreted as pencil by the general sensory area in your right parietal lobe. Your right hemisphere probably cannot speak, but its thoughts can be conveyed by way of the corpus callosum to the left hemisphere, which does have speech areas. Your left hemisphere can say that you are holding a pencil. Other aspects of the division of labor of our cerebral hemisphere are beyond the scope of this book, but it is a fascinating subject that you may wish to explore further. Takeaways 5. 1. The medulla and pons work together for what vital function? The cerebellum and midbrain work together to regulate what physical ability? Answer. The medulla and pons regulate respiration. The cerebellum and midbrain regulate balance or equilibrium. Number 2. Feeling hungry or thirsty, what part of the brain is involved in that feeling? What part of the brain can do something about it? Answer. The hypothalamus is involved in both hunger and thirst. Getting something to eat or drink requires the thinking part of the brain, the cerebrum. 3. State a function of each of these: occipital lobe, frontal lobe, temporal lobe, corpus callosum. Answer. Occipital lobe, vision; frontal lobe, voluntary movement; temporal lobe, hearing or smell; corpus callosum connects the cerebral hemispheres. Meninges and cerebrospinal fluid. The connective tissue membranes that cover the brain and spinal cord are called meninges. 3 layers are illustrated in Figure 8.9. The thick outermost layer made of fibrous connective tissue is the dura mater, which lines the skull and vertebral canal. The middle arachnoid membrane, in parentheses, arachnids are spiders, close parentheses, is made of web-like strands of connective tissue. The innermost pia mater is a very thin membrane on the surface of the spinal cord and brain. Between the arachnoid and the pia mater is the subarachnoid space, which contains cerebrospinal fluid, the tissue fluid of the central nervous system. Recall the ventricles cavities, in parentheses, cavities, close parentheses, of the brain. 2 lateral ventricles, the 3rd ventricle and the 4th ventricle. Each contains a choroid plexus, a capillary network that forms cerebrospinal fluid from blood plasma. This is a continuous process, and the cerebrospinal fluid then circulates in and around the central nervous system. From the lateral and 3rd ventricles, cerebrospinal fluid flows through the 4th ventricle and then to the left central canal of the spinal cord and to the cranial and spinal subarachnoid spaces. As more cerebrospinal fluid is formed, you might expect that some must be reabsorbed, and that is just what happens. From the cranial subarachnoid space, cerebrospinal fluid is reabsorbed through arachnoid villi into the blood and craniovenous sinuses, in parentheses, large veins within double-layered cranial dura mater, close parentheses. The cerebrospinal fluid becomes blood plasma again, and the rate of reabsorption normally equals the rate of production. Because cerebrospinal fluid is tissue fluid, one of its functions is to bring nutrients to CNS neurons and remove waste products to the blood, and as the fluid is reabsorbed. The other function of cerebrospinal fluid is to act as a cushion for the central nervous system. The brain and spinal cord are enclosed in fluid-filled membranes that absorb shock. You can, for example, shake your head vigorously without harming your brain. Naturally, this protection has limits. Very sharp or heavy blows to the skull will indeed cause damage to the brain. Examination of cerebrospinal fluid may be used in the diagnosis of certain diseases. Takeaway 6. 1. Using the terms internal and external, describe the location of the dura mater, and the subarachnoid space. Answer. Number 1, the dura mater is internal to the skull. in parentheses, or vertebrae, close parentheses, and external to the arachnoid membrane. The subarachnoid space is internal to the arachnoid membrane and external to the pia mater. Cranial nerves. The 12 pairs of cranial nerves emerge from the brain stem of other pairs of the brain. They are shown in figure 811. The name cranial indicates their origin, and many of them do carry impulses for functions involving the head. Some, however, have more far-reaching destinations. The impulses for the senses of smell, taste, sight, hearing, and equilibrium are all carried by cranial nerves to their respective sensory areas in the brain. Some cranial nerves carry motor impulses to muscles of the face and eyes or to the salivary glands. The vagus nerves, vagus means wanderer, branch extensively to the larynx, heart, stomach, and intestines, and bronchial tubes. Takeaways 7. 1. Which cranial nerves have functions concerned with the eyes? State these functions. Answer. 1. Optic, sense of sight. Oculomotor, trochlear, and adducens. Movement of eyeball, oculomotor. Constriction of pupil. 2. Which cranial nerve has the most extensive distribution? Where does it go? Answer. CSF is the tissue fluid of the CNS and therefore is similar to tissue fluid elsewhere in the body, in parentheses, and also to blood plasma, from which all tissue fluid is made, close parentheses. The autonomic nervous system. The autonomic nervous system, ANS, is actually part of the peripheral nervous system in that it consists of motor portions of some cranial and spinal nerves. Because its functioning is so specialized, however, the autonomic nervous system is usually discussed as a separate entity, as we will do here. Making up the autonomic nervous system are visceral motor neurons to smooth muscle, cardiac muscle, and glands. These are the visceral effectors. Muscle will either contract or relax, and glands will either increase or decrease their secretions. The ANS has two divisions, sympathetic and parasympathetic. Often they function in opposition to each other. As you will see, the activity of both divisions is integrated by the hypothalamus, which ensures that the visceral effectors will respond appropriately to the situation. Autonomic pathways. Autonomic nerve pathways going from the central nervous system to visceral effectors differ anatomically from somatic pathways that led to skeletal muscle effectors. This is a turn back to figure 8.3 and find the motor neuron. This is a somatic motor neuron. Its cell body is the spinal cord, and its axon extends all the way to the skeletal muscle, where its synapses with muscle cells. A somatic motor pathway, therefore, is a one neuron pathway. An autonomic pathway, however, is a two neuron pathway. It consists of two motor neurons that synapse in a ganglion outside the CNS. The first neuron is called the preganglionic neuron from the CNS to the ganglion. The second neuron is called the postganglionic neuron from the ganglion to the visceral effector. The ganglia are actually the cell bodies of the postganglionic neurons. Look at figure A2 as you read the next sections. Sympathetic division Another name for the sympathetic division is thoracolumbar division, which tells us where the sympathetic preganglionic neurons originate. Their cell bodies are in the thoracic segments and some of the lumbar segments of the spinal cord. Their axons extend to the sympathetic ganglia, most of which are located in two chains just outside the spinal column. Within ganglia are the synapses between preganglionic and postganglionic neurons. The postganglionic axons then go to the visceral effectors. One preganglionic neuron often synapses with many postganglionic neurons to many effectors. The anatomic arrangement has physiological importance. The sympathetic division brings about rapid and widespread responses in many organs. Aspects of the anatomy of both A and S divisions, as shown in Figure 8.12, are summarized in Table 8.5. The sympathetic division is dominant in stressful situations, which include anger, fear, or anxiety, as well as exercise. For our prehistoric ancestors, stressful situations often involve the need for intense physical activity, the fight or flight response. Our nervous systems haven't changed very much in 50,000 years. And if you look at Table 8.6, you will see the kinds of responses the sympathetic division stimulates. The heart rate increases vasodilation and skeletal muscle supplies them with more oxygen. The bronchioles dilate to take in more air, and the liver changes glycogen to glucose to supply energy. At the same time, digestive secretions decrease and peristalsis slows. These are not important in a stress situation. Vasoconstriction in the skin and visceral shunts blood to more vital organs such as the heart, muscles, and brain. All of these responses enable our ancestors to stay and fight to get away from potential danger. Even though we may not always have to respond to life-threatening situations during stress, such as watching a movie from the safety of a living room sofa, sympathetic impulses prepare our bodies to do just that. Figure 8.13 depicts all of the organs innervated by the sympathetic division. The nerve pathways are in blue. Parasympathetic division. The other name for the parasympathetic division is the craniosacral division. The cell bodies of parasympathetic preganglionic neurons are in the brainstem and the sacral segments of the spinal cord. Their axons are in the cranial nerve pairs 3, 7, 9, and 10. In parentheses, oculomotor, facial, glossopharyngeal, and vagus nerves, close parentheses, and in some sacral nerves, and extend to the parasympathetic ganglia. These ganglia are very close to or actually in the visceral effector nerve pathway and contain the postganglionic cell bodies with very short axons to the cells of the effector. These ganglia are very close to or actually in the visceral effector and contain the postganglionic cell bodies with very short axons to the cells of the effector. In the parasympathetic division, one preganglionic neuron synapses with just a few postganglionic neurons to only one effector. With this anatomic arrangement, very localized, in parentheses, one organ, close parentheses, responses are possible. The parasympathetic division dominates in relaxed, non-stressed situations to promote normal functioning of several organ systems. Digestion will be efficient with increased secretions and peristalsis. Defecation and urination may occur, and the heart will beat at a normal resting rate. Other functions of this division are listed in Table 8.6. Notice that when an organ receives both sympathetic and parasympathetic impulses, the responses are opposites. Such an arrangement makes maintaining an appropriate level of activity quite simple, as in changing the heart rate to meet the needs of a situation. Notice also that some visceral effectors receive only sympathetic impulses. In such cases, the opposite response is brought about by a decrease in sympathetic impulses. Secretion by the sweat glands is an example. Neurotransmitters. Recall that neurotransmitters enable nerve impulses to cross synapses. In autonomic pathways, there are two synapses, one between the preganglionic and postganglionic neurons, and the second between postganglionic neurons and the cells of the visceral effectors. Look again at figure 8.2 and find the synapses for each division. Acetylcholine is the transmitter released by all preganglionic neurons, both sympathetic and parasympathetic. It is inactivated by cholinesterase in postganglionic neurons. Parasympathetic postganglionic neurons all release acetylcholine at the synapses with their visceral effectors. Most sympathetic postganglionic neurons release the transmitter norepinephrine at the synapses with the effector cells. Norepinephrine is inactivated by either casetyl-O-methyl transferase, COMT, or monoamine oxidase, MAO, or it may be removed by the synapse by reuptake. The sympathetic postganglionic neurons that release acetylcholine are the neurons to sweat glands, and some of those to the smooth muscles in the walls of the blood vessels and skeletal muscles, in parentheses, to bring about vasodilation, close parentheses. Vasodilation, close parentheses. Takeaways 8.1. State the effects of the two ANS divisions of the bronchioles and the intestines. Which division regulates the diameter of blood vessels and the secretion of sweat glands? Answer. Sympathetic impulses dilate the bronchioles and decrease intestinal peristalsis. Parasympathetic impulses constrict the bronchioles to normal and increase peristalsis. Only the sympathetic division innervates blood vessels and sweat glands. 2. Which ANS division usually has the most widespread effects? Answer. The sympathetic division has widespread effects that enable the entire body to respond rapidly to stressful situations. Aging and the nervous system. The aging brain does lose neurons, but the loss is only a small percentage of the total neurons. The formation of synaptic connections is believed to slow considerably as we get older, but this is not the usual cause of mental impairment. In elderly people, some forgetfulness is to be expected. However, as is a decreased ability for rapid problem solving, but most memory should remain intact. Voluntary movements become slower, as do reflexes and reaction time. Think of driving a car, an ability we sometimes take for granted. For elderly people with their slower perceptions and reaction times, greater consciousness of driving is necessary. Recent research has found that the blood-brain barrier also deteriorates with advancing age. The cells that contribute to making brain capillaries less permeable no longer divide as efficiently, and these capillaries allow molecules such as proteins into brain tissue. This may be a factor in the onset of dementia, as the autonomic nervous system ages. Dry eyes and constipation may become problems. Transient hypotension may be the result of decreased sympathetic stimulation, as vasoconstriction. In most cases, however, elderly people who are aware of these aspects of aging will be able to work with their physicians or nurses to adapt to them. Summary. The nervous system regulates many of our simplest and many complex activities. The impulses generated and carried by the nervous system are an example of the chemical level or organization of the body. These nerve impulses then regulate the functioning of tissues, organs, and organ systems, which permits us to perceive and respond to the world around us and the changes within us. The detection of such changes is the function of the same organs, and they are the subject of our next chapter.